Hello everyone and welcome to today's webinar, Solutions for the Culturing, Maintenance, and Characterization of Induced Pluripotent Stem Cells. I am Cassie Saltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sartorius. To learn more, visit www.sartorius.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Daryl Cole, Application Scientist with Sartorius. Daryl, you may now begin your presentation.
Welcome to this webinar on the solutions for the culturing, maintenance, and characterization of induced pluripotent stem cells. My name is Daryl Cole, and I am an application scientist at Sartorius. So today I'll start with an introduction to induced pluripotent stem cells, or iPSCs, then take a look at an overview of the iPSC culture and characterization workflow using 3 Sartorius products. The first of which is the cell selector Flex, which I'll do a brief introduction to, and take a look at isolation and picking of iPSCs using that system. Following that, we'll have a look at the Incucyte live cell analysis system and see how that system can aid in monitoring iPSC growth and morphology. Then we'll take a look at the iQue high throughput screening cytometer and see how that system can characterize iPSCs through marker expression. And finally, we will conclude what we've learned today. So let's begin with an introduction to iPSCs. Induced pluripotent stem cells, or iPSCs, have the ability to differentiate into multiple lineages depending on their type. They're a highly valuable research and clinical tool and can be used to develop models of normal and diseased tissues. So if you take a look at the schematic on the right-hand side of the slide, you can see the journey an iPSC could potentially take. So starting with a totipotent cell type, we have those cells that can produce all the cells in the body, including placenta and embryo. And moving down from that, we have pluripotent cells, which are what our iPSCs are categorized into. And they can produce all of the somatic cells of the human body and also produce themselves as well. So you can look at blood, cardiac, tissue, neural, bone, hepatic as some examples. They can be derived from induced pluripotent stem cells. So what are induced pluripotent stem cells, or iPSCs? Well, iPSCs are stem cells that have been induced from normal tissues such as fibroblasts through the expression of specific transcription factors. This method was discovered in 2006 by Shinya Yamanaka and his team using Oct4, SOX2, KLF4 and c-Myc, and the reprogramming can be done using a few different methods such as Sendai virus, transfection and episomal induction. On the right, the schematic shows a simplified process for the creation of iPSCs from normal tissue. The cells are harvested, in this case a fibroblast, and are induced to transiently express the transcription factors described. From here, you can maintain these cells in specific medium to create an iPSC line and choose to differentiate those cells into numerous tissues using specific media types and growth conditions. Now that we understand what iPSCs are, let's have a look at the overview of iPSC culture and characterization workflow. On this slide, we have an overview of the iPSC culture and characterization workflow. So starting on the left-hand side, we have the cell selector Flex, which is the system that we will be using to pick and isolate cells. Then in the middle, we have the Incucyte, which we'll use for monitoring morphology and confluency of our iPSCs. And then finally, on the right-hand side, at the end of the workflow, we have the iQue for the characterization of marker expression at the endpoint of our experiments. So let's take a close look at the cell selector Flex and the isolation and picking of iPSCs using this system. Here is the cell selector Flex system. On the left-hand side is the cell selector system with the picking arm and the objectives And then on the right-hand side is the computer system that you would need to control the cell selector flex. So what we have is a very precise robotic arm for cell retrieval by aspiration and scraping. And we have automated microscopy with bright field, phase contrast, and epifluorescence. And the system itself is very powerful. We can analyze images, use those to select cells of our choosing, document any images taken throughout the picking process. And we also have the robotic control through the computer itself. So you can set up automated picking systems of specific cell types based on their characteristics, and then leave the system to go away and do all of that picking for you. On this slide, we detail the exchangeable modules for large application versatility with the cell selector flex. So on the left-hand side, you can see the cell selector flex system, and the circled section is the arm or module, and the system will automatically detect which of these modules you have installed at particular points when you're using the system. And on the right hand side, we've got the 3 different modules that can be used with the system. So we've got the single cell picking module, which uses replaceable glass capillaries. In the middle, we have adherent colony picking module, which uses reusable stainless steel tips, which can be autoclaved for sterilization. And then on the right hand side, we have the semi-solid media picking module, which uses disposable plastic tips. So each of these have specific roles, where the glass capillaries can be used for picking single cells or small colonies. The adherent colony picking module is useful for scraping, so any particularly stubborn cell types that you want to scrape off the bottom of a well. And then the semi-solid media picking module is useful when you're, say, selecting organoids that are grown in a 3D matrix such as Matrigel. So to highlight the capabilities of the cell selector flex in isolating and picking iPSCs, we have a couple of case studies to show you here in this webinar, just to give you an idea of the quality of picking with the cell selector flex. So these images here in the middle are of 2 different iPSC colonies. The one on the left has been picked manually, and the one on the right has been picked using the cell selector flex. And they've been stained for propidium iodide just to look at cell death in these specific colonies. So what you can see from the images is that the manually picked iPSCs have far more dead cells stained with propidium iodide than the cell selector flex iPSC colony. This exemplifies the reliability and gentle iPSC picking with the cell selector flex. And as you can see, the iPSCs are healthier when picked using the cell selector flex compared to a manual iPSC pick. An area where the cell selector flex's accuracy is exemplified is in the isolation of iPSCs using the system. So the images we have in the middle here are of the same section of iPSC culture, but on the left we have a differentiated section prior to picking, and then on the right we have that same section after the differentiated section has been removed by the cell selector flex. So you get an idea of the accuracy and specificity of the system, where if you're growing some nice optimal iPSC clones and you find some differentiated sections within those colonies, you can use the cell selector flex to remove those unwanted sections, ensuring the purity of your iPSC clones. Now, if you wanted to do the reverse, where you have some lovely optimal iPSC colonies growing in a plate, but you start to notice a lot of the other areas of your culture are becoming differentiated, you can use the cell selector flex to go in and pick out that individual colony or multiple colonies that are the optimum in terms of pluripotent phenotype, and then you can grow those on in new plates as a more pure iPSC culture. As alluded to before, using the cell selector flex, because of its accuracy and specificity, you can passage the best iPSCs using the system. So the images here show you an iPSC colony growing on a feeder layer, and what we've done here is picked a specific section of the colony So a pluripotent section, and then we've just gone in there and specifically removed that section without disturbing the surrounding cells. And you can then take that more pluripotent section of your colony and grow that on. And so that allows you to successfully pick for passaging or even downstream analysis, such as marker expression analysis later on in your workflow. Now we're going to take a look at the Incucyte live cell analysis system with a brief introduction, and then we'll look at monitoring iPSC morphology using the system. So here's the Incucyte live cell analysis system. So on the right, we've got a picture of what the system looks like and some of the reagents that you can use with the system in the little tubes there. And it gives you an idea of the sorts of flasks and plates that can be used with the system as well. So the good thing about the Incucyte is that you can leave your cells undisturbed and stationary in the incubator during analysis. So you're protecting the cells from being removed from the incubator multiple times in order to take images. So the image there of the Incucyte itself, that isn't what you would normally see, because usually that would be sitting inside an incubator. You also have setup and walk away convenience. So the integrated image analysis software will allow you to set up your analysis and then view your experiments from PCs as long as they're connected to the same network. The system also allows you to improve productivity because it can accommodate multiple users and applications in up to 6 microplates in parallel. And you can acquire and view images over time. As said before, because the system is sitting in an incubator, you can leave your cells in there for long periods of time and take real-time continuous images and then analyze those images. So you can get growth rates, growth curves, all through longer term studies using the Incucyte. Combining the Incucyte live cell analysis system with one of the previously mentioned reagents, we developed some Nuclite rapid red iPSCs, and they were cultured under optimal and non-optimal conditions to create 2 distinct phenotypes. So the images on the right-hand side show you iPSCs grown under optimized conditions at the top, and then at the bottom, iPSCs grown under non-optimized conditions. So the optimized conditions were iPSCs grown in Nutristem HPSC XF. So that's a media produced by Sartorius, developed specifically for iPSC or stem cell growth. And then at the bottom, those same cells were grown in RPMI, so a standard culture media that's lacking in all of the growth factors and cytokines required for iPSC growth. So you can quite clearly see a change in morphology between the 2 conditions. So the cells were monitored for 2 days and then analyzed using the Incucyte live cell analysis system. And as I've mentioned, the morphological difference is clearly visible from the images collected. The optimized iPSCs form densely packed colonies, which you can see from the nuclear staining on the right-hand side, and the non-optimized iPSCs are far more spread out with a less dense colony formation, more indicative of a fibroblastic appearance. And you can again see that with the nuclear staining of those particular cells on the right-hand side at the bottom. Using the Incucyte and the Nuclite red iPSCs, we are able to monitor pluripotent potential in these particular cell types. So we can quantify morphological differences. During this experiment, we used adherence cell-by-cell scan, the basic analyzer, and the AI confluence analysis. So the basic analyzer and AI confluence analysis were used for nuclear and cytoplasm area measurements. So what you can see on the left-hand side in the images are the masking used by the system in order to identify the total area of the confluence and then the individual nuclear area from the Nuclite red staining. So with this information, we created a ratio called the nuclear cytoplasm ratio, which is used as a standard tool for measuring iPSC pluripotency in imaging. And we took the total nuclei area, divided it by the total cytoplasmic area, and that gave us this ratio. And from the graph, also it's very clear in the image itself as well, in the images, but from the graph you can see that the ratio is higher in the optimized iPSCs, which is indicative of a more pluripotent phenotype, where the iPSC colonies are much more densely packed with many more nuclei wedged into a smaller area than in the non-optimized, where the cells are much more spread out and the nuclei have a lot more space between them because the cells themselves have spread out over the plate.
Now we'll take a look at the iQue high throughput screening cytometer. This is the iQue high throughput screening cytometer. It's a high throughput, high content cell analysis solution. It can analyze both cells and beads for antibody screening and also functional profiling. So in the middle of the slide, you can see an image of the iQue there, and it comes with its own proprietary software called the Foresight software. And you can detect cell by cell, so thousands of cells per second with multiple readouts for every single event. And you can analyze the natural conformation of cells and targets. Because of the design of the system, you can save precious samples. The sample sizes go down all the way down to 1 microliter per sample. So you can save any samples that you may want to use downstream. It's user-friendly as well, so there's no dead volume. The entire sample is taken to the detector. It's very, very fast, so you can answer more questions in less time. A 384 well plate is measured in around 15 minutes, and a 96 well plate can be measured in less than 5 minutes.
Using the iQue advanced flow cytometer, we can characterize iPSC pluripotency and phenotype via marker expression analysis. So for this particular experiment, we took 2 intracellular markers, OCT3 and 4, and SOX2. These are markers of pluripotency, and then we combined them with 3 cell surface markers, SSEA1, a marker for normal non-pluripotent cells, and then SSEA4 and Tron60, 2 markers of pluripotency. We combined this with a fixable viability dye, so we could measure our live cells, and perform this work in 96 well plates with iPSC stained with all the markers and the viability dye in the same well in a multiplexed experiment. On this slide, we can see the data that we gathered using the iQue to measure surface and intracellular marker expression in our iPSCs. So what we have here are some histograms on the top row and some dot plots on the bottom row. And these 2 ways of presenting the data are derived directly from the Foresight software that you would use with the iQue. So we have in yellow THP1s, which are a normal cell control. And then in black, we have iPSCs that we've been growing for this experiment. And if you look over onto the right side of the slide, you can see that we have 2 markers, SOX2 and OCT3 and 4. These are our intracellular markers. And you can see that the expression is only found in our iPSCs. The THP1s have no expression. You see that shift over to the right in the iPSCs, suggesting increased expression of SOX2 and OCT3-4, whereas in the THP1s in yellow, the shift is to the left for the peaks and also the dots in the dot plot. And that indicates that there is no expression of these iPSC markers for pluripotency. We also have on the far left SSEA1 marker expression, and that's marker expression for non-pluripotent phenotypes. And what we can see here is that we have the reverse to the intracellular markers. So our iPSCs have no discernible expression of SSEA1, whereas our THP1s have a high expression of SSEA1. And then to the right of that particular set of histograms and dot plots, we have SSEA4 and Tron60. And these are the surface markers for iPSC pluripotency. And again, we can see similarly to the intracellular markers, we have high expression in our iPSCs and no expression in the THP1 control cells. So both these histograms and dot plots were created in iQue Foresight, and the particular populations you see here are gated from live cells, so this is just live cell expression of these particular markers. So what we've shown here is that using the iQue, we can characterize our iPSCs for 4 different pluripotency markers, and then also look at a normal cell marker in SSEA1. and compare the iPSCs to a normal control cell in THP1s. Combining this data into a couple of figures here highlights the flexibility of the Foresight software and allows us to visualize the data more clearly. On the left is a combined heat map with each marker pulled out to easily distinguish the expression pattern. As mentioned previously, this experiment was a multiplex assay, so each cell line was stained for all markers in each well in a total of 4 wells, and the heat map has just had those individually mapped here for visualization purposes, with yellow indicating lower expression levels and black indicating higher expression levels. On the right is a bar graph illustrating the different expression patterns between the cell types, where we can see the complete marker expression. THP1 cells in yellow are clearly expressing SSEA1 non-pluripotency marker and not expressing the pluripotency markers, while the iPSCs in black are expressing all pluripotency markers with no expression of the non-pluripotency marker. This data exemplifies the benefits of using the iQue platform for surface and intracellular marker phenotyping of precious cell types such as iPSCs for cell line development and other workflows where flexible, high-throughput, fast and robust analysis is required for important decision-making throughout such workflows. A further benefit is the small sample requirements of the iQue, saving precious sample for downstream processes. In a similar vein to the data shown in the Incucyte section of this webinar, here we have some iPSC pluripotency marker characterization on the iQue for iPSCs grown in optimized and non-optimized conditions. So as before, the iPSCs grown in optimized conditions were grown in stem cell specific media, specifically Nutristem, and the non-optimized iPSCs were grown in RPMI. We have the same control in the THP1s as our normal controls, but we also have NCCI T cells, which act as a pluripotent cell control, so they express pluripotent markers and do not express the non-pluripotency marker SSEA1. So what we've got here are iPSCs cultured in optimized and non-optimized conditions, and then surface marker expression analyzed on the iQue. So if you take a look at the graph at the top on the right hand side of the slide, you can see that our THP1s have high levels of SSEA1, the non-pluripotency marker, and no expression of the pluripotency markers SSEA4 and Tri160. And just to explain the pluripotent portion of the graph, the grey bars, they are all cells that are SSEA1 negative but double positive for SSEA4 and Tri160. So that's our pluripotency measure for our iPSCs. And if you look below that graph, you can see the dot plots that give us an idea of the changes in marker expression in optimized and non-optimized conditions. So on the optimized side, you can see there are no expression of SSEA1, but then the cells that are grown in the non-optimized conditions, you see a shift over to the right where they're beginning to express SSEA1. So comparing the two, iPSCs in the optimized conditions have high levels of pluripotency markers and no expression of the non-pluripotency marker, whereas the iPSCs in the non-optimized conditions have reduced levels of pluripotency markers and an increased expression of the non-pluripotency markers. So this is a nice way of showing how the iQue can confirm some of the data that we have already gathered on the Incucyte. So you can run your cells through the Incucyte, perform all of that monitoring of morphology, and then you can collect those cells at the end and run them through the iQue to confirm your analysis there. On the next couple of slides, I'll summarize what we've learned today. So let's discuss what we've learned today. What we've shown is that we can reliably pick and isolate optimal iPSC colonies using cell selector flex. Because the system is gentle, it provides highly viable iPSC cultures for passaging and downstream processing. And due to the system's high levels of accuracy, you can target specific regions of iPSC colonies for isolation and picking. Using the Incucyte live cell analysis system within this workflow, you can monitor morphology and pluripotent potential. So iPSC cultures can be monitored over time in a non-perturbing manner within the incubator environment. Using Incucyte analysis software, you can assess iPSC pluripotent potential as well. And using the iQue HTS cytometer, you can characterize and analyze iPSC phenotype and pluripotency status. Multiplexing experiments can provide flexibility for characterization of multiple metrics, such as surface and intracellular marker expression within the same well using the iQue. And the iQue Foresight software provides simple and powerful analysis of iPSC marker expression data. So if we take another look at the workflow schematic here at the bottom of the slide, you can see that we can use various Sartorius platforms throughout an iPSC culture workflow. We can pick and seed using the cell selector flex, and then take those cells, monitor their morphological status using the Incucyte, and then from there, harvest those cells and characterize their pluripotency through endpoint analysis on the iQue, allowing you to have a fully rounded iPSC culture workflow using these three systems. If you'd like any further information on the systems that I've presented today, please see these web pages. We would now like to take some time to answer any questions that the audience may have. Thank you for your attention today. Please feel free to reach out at the email on the screen with any additional questions you may have.
Thank you, Daryl, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the lower left corner of your screen. We'll answer as many questions as we have time for. Let's go ahead and get started here. Our first question asks, can you image fluorescence using the cell selector, for example, when isolating and picking fluorescently labeled iPSCs?
So yes, cell selector flex is capable of imaging in multiple different fluorescent channels, so it's more than capable of performing that kind of experiment.
Excellent, thank you. Moving along here, our next question asks, is it possible to pick organoids with the cell selector?
Again, yes, the cell selector flex can pick organoids and other 3D structures such as spheroids or iPSC spheroids from media and 3D matrices such as Matrigel.
Great, thank you. Our next question here asks, how is the iQue different from a standard flow cytometer?
So one of the main differences with the iQue versus a standard flow cytometer is the way that it actually processes the samples. So it uses an air gap solution, so it can more quickly process your 96 well plates. So essentially what it does is it takes your sample, then it places an air gap between it, and then it can also pick another sample and do that throughout the plate, rather than having to stop, do the picking of the sample, process it at the same time. It can then have multiple samples running through the system at the same time.
Okay, great, thank you. Another question here asks, how can we visualize stem cell differentiation using your analyzer?
I'm guessing you mean the iQue by the analyzer. We've done this using a multitude of different stem cell or differentiation markers. So what we would do in a very similar vein to what we've talked about in the webinar, you would take your cells during the differentiation process and you would stain them for different markers that you would expect to see during that differentiation. And then you can analyze that and you can ensure that your cells are being directed towards the correct lineage that you're looking for.
Excellent, thank you. Our next question here asks, can the Incucyte measure growth of iPSCs over time?
So yeah, with that one, the Incucyte can allow users to measure iPSC confluency over time, as I showed briefly in the webinar, and that allows you to determine changes in growth patterns in different conditions. Okay,
wonderful. And we have time for one more question here. This question asks, how difficult is it to dissociate iPSCs for analysis on the iQue?
In our hands, iPSCs can be fairly easily dissociated in a few minutes using dissociation reagents such as Accutase, and then we'd stain for marker expression for analysis in the iQue. The most important thing is to make sure you've got a single cell solution at the end when you do your staining.
Okay, and we had one more question slip in under the radar, so I'm going to go ahead and ask that one. It says, what markers and cells would you use to show that a pluripotent stem cell is undergoing dedifferentiation into a tumor-specific phenotype? For example, in response to a pharma small molecule or biologic treatment. So I'm asking about embryonic phenotyping that results in developmental toxicology.
So for that, I mean, I would use iPSCs in that particular experiment. In terms of markers, I wouldn't be able to go into specifics of that, but I'd be happy to answer that another time.
Okay, great. And we did have a couple questions come on in, so if you don't mind, we'll keep asking them.
Yeah, that's okay.
Okay. This next question here says, could you please explain more about the mechanism of action?
I'm not entirely sure what mechanism of action that question relates to, so I think I need a bit more information to answer that.
Not a problem. If you want to go ahead and submit a follow-up question with more details, we can try to get to that during this webinar. We'll move on to our next one here. It says, what are the effects of different culture media formulation on iPSC population?
So the example that I showed during the webinar, we get changes in morphology and also a reduction in the iPSC growth. So iPSCs tend to grow fairly quickly, and they, due to their pluripotent nature, you're able to keep growing them much like a cancer cell line. Whereas the cells that undergo the differentiation that we've shown in the non-conditioned or the unconditioned media type, those cells begin to stop growing, and they will stop. They'll stop propagating any further. So.
Okay, thank you. I don't see the follow-up in here, so we'll go ahead and wrap with that final question. I want to thank you for your time today and your important research. We would also like to thank LabRoots and our sponsor, Sartorius, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during our on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.